Hello, I am Jung Shin from South Korea, and I'm currently a researcher at Sungkyunkwan University. And I'm about to start my PhD degree at UT Austin from this fall. And today I'm going to talk about my recent research entitled Non-invasive cell tracking system using lipid-supported polymeric nanoparticle. As you might know, cell tracking is kind of very important technology when doing the cell-based therapy because we have to know the transplanted cell destination, or we have to prevent some side effects caused by the transplanted cells. So today I want to talk about the cell tracking system that I designed. Okay, so first I'm going to talk about the introduction of this technology for your easy understanding, and I'm going to show you some experimental results. And the results will contain light-emitting polymeric nanoparticle and light-emitting cells using cell surface modification method and some preliminary in vivo feasibility for cell tracking. Okay, so this is the conventional cell tracking method. When doing the stem cell-based therapy or immune cell-based therapy, we have used PET or CT, MRI, or such technologies to track the transplanted cells. In some cases, in small animal study, we have used fluorescence imaging method, but as you might know, fluorescence imaging has limitation because it has very short tissue penetration ability. So as a non-invasive cell tracking method, near-infrared imaging has a very good potential to be used in cell tracking. NIR region has some advantages because they have deep tissue penetration ability, and the technology is cost and time effective, and it has good accessibility. Also, NIR window is does not disturbed by H2O or hemoglobin, which is very abundant in our body, and can be an obstacle when imaging the cells. However, in order to see the transplanted cell in NIR region, the transplanting cell should be labeled with NIR dye or NIR dye containing nanoparticles. So there are two conventional methods to label cells, which are direct labeling method and indirect labeling method. Direct labeling method means we need to put the dye or nanoparticle inside the cells, which means the molecules will be penetrating inside the cells. And indirect labeling method means we should transfect the genes to inhibit some light. And you can guess there are disadvantages to use these conventional methods because they will directly affect the cell metabolism, and some studies says the transfection method can cause some carcinoma. So, and we have to use the cells for therapeutic effect. So maintaining the intrinsic functions of the cells would be very important. So we have referred a paper which is published in 2010 in Nature Medicine by Detter J. Urban Group in MIT. And they have conjugated the nanoparticle to the cell surface for prolonged therapeutic effects. So there are some free Shire groups at the surface of the cell membrane, proteins, and they have used the malamide group-coated nanoparticles so that the nanoparticles can be attached to the free Shire groups at the surface of the cell. So this is our system that we have devised, and this is the scheme of our particle. We have encapsulated indocyanine green NIR dye, which is the FDA-approved dye, inside the PLGA nanoparticle, and we have coated the nanoparticle with lipid bilayer. And the bilayer has composition of DOPC, DOPG, and MPBPE, And at the head of the MPBPE, there are malamide groups. So that is, the total surface of the nanoparticle will be coated with the malamide group. So this is the SEM image of the nanoparticle. And we could see that this is spherical, and we could measure the hydrodynamic diameter using the DLS, and it was measured to be about 156 nanometer. And this particle was stable, quite stable, up to 14 days when measured the hydrodynamic diameter through the DLS. And then we have observed the NIR image of the nanoparticle dispersed solution. And when we see the light-emitting polymeric nanoparticle, we call this LEMP. And this one is the polymeric light-emitting particle, and this one is the empty nanoparticle. So we could see the nanoparticle dispersed solution showed the NIR signal. And then we have conjugated the nanoparticle onto the surface of the cells, and we have used two kinds of cells. The one is human umbilical cord blood-derived mesenchymal stem cells, and the other one is naive CD4 positive T cells, and we call this we call this stem cell light-emitting LESC, and the T cell LETC. When compared the LESC and using the same amount of ICG, free ICG, that is, the free ICG was uptaken by the stem cells, and when using the light-emitting nanoparticle, the cell viability was over 90%, but when using the free ICG, it showed about 80%. So it means when using the nanoparticle, it reduced the cell cytotoxicity. Also, we have analyzed the dose-dependent cell viability assay using two kinds of cells, and there were no big effects on the dose. And then we have determined the amount of the LEMP dose through the flow cytometric analysis. In case of using the stem cells, the mean fluorescence intensity value was saturated from 4 milligram, and it showed the same trend with the CD4 positive T cells. So we have determined to use the 4 milligram of LEMP per 4 times 10 to the 5th cells for both in vitro and in vivo experiment. And we have observed the morphology of the cells, and we have confirmed the signal of the nanoparticle was shown at the surface of the cells. Both are the stem cells, and this is the stem cells. And this is the same stem cells, but the stems are not stretched out. And this is the CD4 positive T cells, but in case of T cells, the cells are too small, so we cannot observe the exact image like stem cells. And then to confirm whether the cells are maintaining its functionality, we have done some experiments, and we have analyzed CD markers at the surface of the human MSC. And we have found out that human MSC's negative markers and positive markers does not have difference with the control group, which is the native human UCB MSCs. And we have done the migration assay, and as you might know, migration ability is quite important in case of both stem cell and T cells, but in case of stem cells, there are harming effects in response to inflammation or something like that. So we have compared the migration ability, and there was also no difference between the control group and the LESC, and the migration ability was about 46%. And differentiation assay, which might be the most important functionality in stem cell, is ongoing. And we have done preliminary in vivo feasibility test. We have injected 8 times 10 to the 5th of LESC in intramuscularly and observed the signal, NIR signal of the mouse from the NIR camera. And we could confirm that the signal is maintained up to 14 days, and the intensity was decreased in exponential function. And when you see this graph, control means using the free ICG labeled MSC. And at the day 14 from the injection, the signal was higher in case of LESC than the control group. And when each organs were dissected at the day 14 from the injection, we could observe some NIR signal at each organs. And then we have injected the dose-dependent, the different amount of LESCs in the same way. And we could also confirm they all show the exponential decrease, and we could regress the equation like this. So yeah, and it showed quite high reliability in the regression equation. So as conclude this study, this system would be feasible in tracking various cell types because cells has the membrane proteins, and membrane proteins has the free silo groups. And this system does not affect cell functionality so that the cells could be utilized for cell-based therapy. And of course, this study is kind of preliminary study, so it needs to be reinforced for a more prolonged duration or more stable system. Okay, so thank you for your attention.